Welcome back to the deep dive. This is where we take your stack of sources, the articles, the research, the notes you shared, and while we plunge right in, we unpack the complex stuff, find those hidden connections, and distill the really important insights.
Exactly. Think of us as your guides, navigating through sometimes dense information. Our goal is to help you cut through it all and, walk away feeling genuinely well-informed, maybe with those aha moments that make learning stick.
Precisely. And today, we've got some incredibly detailed source material. It looks like excerpts from a pretty foundational physiology text. The topic is fundamental, but the level of detail is anything but basic. We're taking a really deep dive into male reproductive functions.
That's right. This material, it lays out everything, the anatomical structures, the complex cellular journeys, the whole intricate dance of hormones, and the physiological mechanics behind it all. It's the kind of depth that, honestly, can feel a bit overwhelming at first glance.
So our mission today is to explore these pages together. We want to pull out the essential processes, really understand the why behind the biological how, and put it all into context. By the end of this deep dive, you should feel like you truly grasped these core functions.
Yeah, we'll unravel the fascinating timeline of sperm development, uncover how hormones act like these molecular messengers, explore the surprising mechanics of the male sexual act, and even touch on, the potential implications when things don't quite go according to plan. Get ready to see the biological detail really up close.
All right, let's jump right in. Let's start with the foundational plumbing, as it were, the physiological anatomy of the male sexual organs.
Okay. To really understand how the whole system works, we first need a clear picture of the components and how they're all connected. The text provides a great map, actually, especially with figures like 81-1A and 81-1B showing the layout. It's a surprisingly intricate setup.
It really is. And the heart of it all, it seems, is the testes. What's its primary role, according to the text?
So the testes is basically the factory for sperm production. Inside each testes, the text tells us, there are up to 900 tiny, highly coiled tubes. These are the seminiferous tubules.
900. Wow, that's a lot. And they're not short tubes, are they?
No, far from it. Each one is over half a meter in length. So just picture this enormous length of microscopic tubing, over 450 meters total in each testes, packed and coiled up tightly. This is the actual site where spermatogenesis, the formation of sperm, takes place.
Incredible. And once sperm are formed in these tubules, where do they go next? What's the path?
They move from the seminiferous tubules into another structure called the epididymis. This is essentially a continuation of the system described as a single, very long, coiled tube.
How long are we talking for the epididymis? Is it comparable?
Well, the text puts it at about 6 meters in length. 6 meters. So sperm embark on quite a journey just to get through this initial storage and maturation area before they move on.
6 meters of coiled tube attached to over 450 meters of other coiled tubes. The sheer scale packed into one testis is, well, it's kind of wild when you think about it.
It certainly is. From the epididymis, the pathway continues into the vas deferens. This tube travels upwards, and right before it reaches the prostate gland, the text notes it enlarges slightly into what's called the ampulla of the vas deferens.
Okay, so we've got the testis, the epididymis, the vas deferens, and its ampulla. Where do the fluids that make up semen actually get added into the mix?
Ah, right. That's where the accessory glands come in. The seminal vesicles, there are 2 of them, one on each side. They're located behind the prostate. And the text says they empty their secretions into the prostatic end of the ampulla of the vas deferens. So sperm from the vas deferens and fluid from the seminal vesicles mix right there.
And that combined stream then flows into the ejaculatory duct. Is next?
Precisely. The ejaculatory duct is formed by the joining of the duct from the seminal vesicle and the ampulla of the vas deferens. And the key anatomical point here is that this ejaculatory duct then passes directly through the substance of the prostate gland itself.
Meaning the prostate literally surrounds the pathway at this crucial point.
Yes, it sits right around the ejaculatory duct and also the initial part of the urethra, which is called the prostatic urethra. The prostate gland itself also has ducts, and these empty its own fluid into both the ejaculatory duct and directly into the prostatic urethra.
So everything kind of converges there before entering the main urethra.
Exactly. The urethra is the final common pathway. It receives the mixture from the ejaculatory ducts and the prostatic ducts, then travels through the penis, and serves as the exit route for both semen and urine from the body.
The text also mentions some smaller glands contributing mucus, doesn't it, for lubrication?
That's correct. There are numerous minute urethral glands scattered along the length of the urethra itself. And then, located near the very origin of the urethra, are the more prominent bilateral bulbourethral glands, also known as Cowper glands. These secrete mucus, which contributes to lubrication during intercourse.
Okay, so stepping back, it's this whole sequential system. Formation in the seminiferous tubules, passage and initial storage in the epididymis, transit through the vas deferens, mixing with seminal, vesicle, and prostatic fluid in the ejaculatory duct and urethra, and finally the urethra carrying the resulting semen outwards.
It's a complex network, yeah. Ducts and glands, each with a really specialized role in transporting sperm and adding necessary components to the semen. Understanding the structure as well, it's foundational to appreciating the function.
It certainly is. And speaking of function, let's get into the process of sperm formation itself. The text gives this a dedicated section, calling it spermatogenesis.
Spermatogenesis.
Right.
This is the remarkable process by which the male gametes, the sperm, are actually produced. And as we just noted, this happens within those incredibly long, highly coiled seminiferous tubules inside the testes.
And this isn't a process that's running from birth, is it? It starts later?
No, it begins at puberty. The text puts the average age at around 13 years old. But once it starts, it continues throughout most of the man's life, although the rate does tend to decrease somewhat in old age. We'll see later that this whole process is critically dependent on stimulation by gonadotropic hormones from the anterior pituitary gland.
Where do the cells that eventually become sperm actually originate? Do they start in the testes?
Well, the journey starts even earlier, during embryonic development, actually. Primordial germ cells migrate into the testes. Once they're there, they develop into what are called immature germ cells, known as spermatogonia.
And where are these spermatogonia located initially? Within the tubules?
They sit in layers, usually 2 or 3 deep, right along the inner surface of the seminiferous tubule wall. The text highlights this in figure 81-2a, showing how these spermatogonia are kind of nestled within the supporting structure provided by much larger cells called Sertoli cells.
So they're present in childhood, but essentially dormant.
Inactive, that's right. They're there, but relatively inactive. Puberty is the trigger. The text describes that at puberty, under hormonal influence, these spermatogonia begin rapid mitotic division. This is a proliferative stage, basically creating many copies of themselves. Then they begin to differentiate, moving through a very specific series of stages depicted in figure 81-2b.
So it's a clear progression starting from those initial spermatogonia.
Absolutely. The text lays out the key steps, also illustrated nicely in figure 81-3. The spermatogonia first migrate among the Sertoli cells, moving inwards towards the central lumen, the open channel of the tubule. The Sertoli cells are crucial support players throughout this entire process. They're these large cells that literally surround the developing germ cells with their cytoplasmic envelopes, nurturing them.
What happens as the spermatogonia move closer to the lumen? Do they change?
Yes. Spermatogonia that cross a certain barrier among the Sertoli cells become modified and significantly enlarge. At this point, they are called primary spermatocytes.
And these primary spermatocytes are the ones that undergo meiosis?
That's the next critical step. The primary spermatocytes undergo the first meiotic division. Now, this is a reductive division where the chromosome number is halved. Each primary spermatocyte, which started with the full complement of 46 chromosomes, 23 pairs, divides to form 2 secondary spermatocytes, each containing only 23 chromosomes.
And then those secondary spermatocytes divide again?
Yes, after a short interval, often just a few days, each secondary spermatocyte undergoes a second meiotic division. This results in the formation of spermatids. So from a single primary spermatocyte, you ultimately get 4 spermatids, and each of those has 23 chromosomes.
This is where the genetic diversity gets created, right?
Yeah.
Having the chromosomes and mixing them up during meiosis.
Precisely. During these meiotic divisions, not only is the chromosome number reduced by half, but there's also genetic recombination, shuffling the deck, so to speak. This ensures that each spermatid is genetically unique, carrying a different mix of genes inherited from the individual's mother and father.
So you end up with these spermatids, but they aren't functional sperm yet. They need more work.
No, they're not.
Oh.
They're sort of like the precursors. The spermatids are then transformed through a process of differentiation into the mature spermatozoa, which are the motile sperm cells we typically picture. This final transformation process is sometimes specifically called spermatogenesis. The text notes that FSH, one of those pituitary hormones, is particularly important for this specific differentiation step.
The entire process, from the initial spermatogonia all the way to the final spermatozoa, takes a considerable amount of time, according to the text.
It does. The text states the entire sequence takes about 74 days. 74 days. This means that at any given time, the seminiferous tubules contain a mix of germ cells at all these different stages of development, from spermatogonia on the periphery near the wall to fully formed sperm near the central lumen, ready to move on.
Let's circle back to the sex chromosomes during those meiotic divisions. How does that determine the potential sex of offspring?
Right. So in each spermatogonium, the sex is determined by one of the 23 chromosome pairs, specifically the X and Y chromosomes. Males have one X and one Y. During meiosis, when the chromosomes are separated into the spermatids, one spermatid receives the Y chromosome. This will develop into a male sperm. The other spermatid receives the X chromosome. This develops into a female sperm.
So the sex of the future offspring is entirely dependent on whether an X-carrying sperm or a Y-carrying sperm happens to be the one that fertilizes the ovum.
That's the core mechanism of sex determination from the male side, yes. If an X-carrying sperm fertilizes the ovum, which always carries an X chromosome from the mother, the result is XX, a genetic female. If a Y-carrying sperm fertilizes the ovum, the result is XY, a genetic male.
Incredible how that fundamental aspect of inheritance is built right into the meiotic process. Now, tell me more about that final transformation, spermatogenesis. The text shows it in figure 81-4. What does the spermatid actually become? What changes?
The spermatid undergoes this really dramatic elongation and restructuring to become the characteristic spermatozome. The head is the most critical part, obviously, containing the highly condensed nucleus with all the genetic material covered by just a thin cell membrane.
And that cap-like structure on the head, the acrosome, what's that about?
Yes, the acrosome. It forms as a cap covering the anterior or front two-thirds of the sperm head. Interestingly, it's derived from the Golgi apparatus of the original spermatid cell. This is a really vital component because it contains potent enzymes.
What kind of enzymes are we talking about, and what do they actually do?
The acrosome contains enzymes similar to those found in lysosomes and other cells, notably hyaluronidase and various proteolytic enzymes. Hyaluronidase is crucial for breaking down hyaluronic acid polymers, which are like cement in the ground substance of tissues. The proteolytic enzymes digest proteins. Basically, these enzymes are the biological tools the sperm uses to penetrate the protective layers surrounding the ovum during fertilization.
So it's kind of like a biological drill bit filled with dissolving agents.
That's a pretty good analogy, yeah. The enzymes allow the sperm to digest a pathway through the protective layers around the egg, essentially clearing the way.
And the tail obviously gives it mobility. That's the propeller.
Exactly. The tail, or flagellum, is the engine. It has three main parts. A core skeleton, called the axonome, composed of 11 microtubules arranged in a specific pattern, similar to those found in cilia, a thin cell membrane covering the axonome, and then a collection of mitochondria concentrated in the upper part of the tail, the body of the tail, surrounding the axonome.
The mitochondria provide the power source, ATP.
Yes, they produce ATP, the energy currency for the tail's movement. The text describes the motility as a flagellar movement, a rhythmic, whip-like motion caused by the longitudinal sliding of the microtubules within the axonome structure.
How fast can they actually swim?
A normal, healthy sperm can move at a velocity of about 1 to 4 millimeters per minute in fluid. It might not sound incredibly fast in our terms, but considering the tiny scale of the sperm itself, it's quite efficient propulsion.
So spermatogenesis is this incredibly complex 74-day journey within the seminiferous tubules, starting at puberty, involving intricate cell division and differentiation under hormonal control, resulting in this highly specialized cell with genetic cargo, an enzyme-filled head for penetration, and a power-generating tail for movement. That's just an incredible transformation.
It really is a masterpiece of biological manufacturing. But interestingly, even after this detailed formation process, the sperm aren't immediately ready to fertilize an egg. They still need to undergo further changes after leaving the tubules.
Ah, okay. Which brings us to the next stage then. Maturation, storage, and the physiology of sperm after they leave the seminiferous tubules.
Right, and a critical point from the text here is that sperm, as they emerge from the seminiferous tubules, are actually non-motile. They can't swim yet, and they're incapable of fertilization. They need some post-production processing.
And that happens in the epididymis, that really long coiled tube we talked about.
Primarily, yes. It takes several days for the sperm to transit through the entire 6-meter length of the epididymis. During this passage, they develop the capability of motility.
The capability. So they still aren't swimming vigorously inside the epididymis itself.
No, they're not. The fluid within the epididymis contains specific inhibitory proteins that actually suppress the sperm's final motility. This inhibition is crucial for storage. essentially preventing them from expending all their energy prematurely before they're needed.
Speaking of storage, given that incredibly high production rate you mentioned, where are they kept?
Yeah, the text notes that an adult testes can produce up to 120 million sperm per day. That's a huge number. The majority of these are stored in the epididymis, with a smaller quantity potentially accumulating further down the line and thus deferens.
And how long can they be stored there while still remaining fertile?
They can stay stored in this suppressed fertile state for at least a month, maybe longer, thanks to those inhibitory substances in the duct secretions keeping them quiescent. However, if there's a high level of sexual activity with frequent ejaculations, the storage duration is obviously reduced, maybe down to only a few days.
So the epididymis acts as a storage facility where they gain the potential for motility, but that potential is kept under wraps, inhibited. What happens when they are finally ejaculated?
Okay, so upon ejaculation, the sperm become fully motile and capable of fertilization. The text uses the term maturation here again, sometimes specifically referring to the changes in the female tract as capacitation, which we'll detail shortly. But the key is this post-ejaculation activation.
And the fluids ejaculated along with the sperm play a role in this activation.
Yes, definitely. The Sertoli cells back in the tubules and the epithelium lining the epididymis secrete a special nutrient fluid. This fluid contains things like hormones, testosterone, and estrogens, interestingly, plus enzymes and other nutrients. This fluid is ejaculated along with the sperm and contributes to their nutrient supply and the overall activation environment.
Let's talk more about the factors affecting mature sperm physiology. You mentioned pH earlier being important.
Yes, pH is critically important for sperm motility. Their activity is significantly enhanced in a neutral or slightly alkaline medium, like that of ejaculated semen, which has an average pH around 7.5. Conversely, even mildly acidic conditions depress their activity, and strongly acidic fluid can rapidly kill them.
Where would they normally encounter such acidic conditions?
Well, within the male reproductive tract itself, the fluid in the vas deferens can become slightly acidic due to the sperm's own metabolic activity producing acidic byproducts. But critically, in the female reproductive tract, vaginal secretions are normally quite acidic, typically with a pH between 3.5 and 4.0. This is why the alkaline fluid contributed by the prostate gland, which gets added to semen during ejaculation, is so absolutely vital. It helps neutralize both the slight acidity from the vas deferens and, more importantly, the significant acidity of the vagina, bringing the overall semen pH into the optimal range of about 6.0 to 6.5 for sperm motility and fertility.
So the alkaline prostate secretion is really a fertility enhancer just by adjusting the pH, creating a more welcoming environment.
Absolutely. It's a key component for creating the right chemical environment, especially in the initial phase within the female tract.
Temperature also affects them, right? We touched on the scrotum's role.
Yes, their metabolic activity and motility increase with temperature, but this also shortens their lifespan because they burn through energy faster. While they can survive for weeks in the male ducts in that suppressed state, their lifespan in the female tract at body temperature is quite limited, typically only 24 to 48 hours, according to the text.
But we know they can be stored for much longer outside the body for fertility purposes, right? Like sperm banking.
Yes, the text confirms this. At lower temperatures, they can be stored for weeks. And if frozen at temperatures below -100 degrees Celsius, they can remain viable, potentially fertile, for years. This is the basis of cryopreservation used in sperm banking and assisted reproduction.
So sperm undergo this critical maturation and storage phase in the epididymis, remain suppressed until ejaculation, and then rely heavily on the pH and nutrient content of the ejaculated fluid, as well as temperature conditions, to become fully active and survive the limited time they have in the female tract.
It's a really tightly regulated transition from quiescent storage to active searchers. And speaking of the ejaculated fluid, let's look closer at the function of those accessory glands, the seminal vesicles in the prostate, and the specific fluids they contribute.
The seminal vesicles add a large volume, don't they? What's in their fluid?
They do contribute a significant volume. The seminal vesicles are described as these tortuous, loculated tubes lined with a secretory epithelium that produces a mucoid material. The text lists its key components: a significant amount of fructose, citric acid, other nutrient substances, large quantities of prostaglandins, and also fibrinogen.
Fructose for energy, presumably. And this fluid is added during emission.
Yes, exactly. Fructose is a key carbohydrate that provides a readily available energy source for the ejaculated sperm, powering their motility as they make their journey. And yes, during the process of emission and ejaculation, the seminal vesicles empty their contents into the ejaculatory duct shortly after the vas deferens has contracted to expel the sperm.
How much volume does this seminal vesicle fluid actually contribute to the final semen?
The seminal vesicle fluid makes up the largest portion. The test says almost 60% of the total semen volume. And because it's added relatively late in the emission sequence, it effectively helps wash the sperm through the ducts and the urethra.
Okay, so bulk and energy. What about the prostaglandins? The text suggests they have a couple of interesting potential roles related to aiding fertilization.
Right. The text proposes two potential roles for the prostaglandins from the seminal vesicles in assisting fertilization. First, they are thought to interact with the cervical mucus in the female tract, making it thinner or more receptive to sperm movement, easier for sperm to penetrate. Second, they may cause reversed peristaltic contractions in the uterus and fallopian tubes.
Reverse contractions. In the female tract, that sounds like it would actively help pull or propel the sperm upwards towards the egg.
That's the hypothesis presented in the text, yes. This possible action might help explain how some sperm can reach the upper parts of the fallopian tubes relatively quickly. The text mentions within as little as 5 minutes, which is much faster than their own swimming speed would allow over that distance.
That's a fascinating potential interaction. Male-produced molecules influencing female physiology to aid transport. So seminal vesicles provide bulk, energy, and these potentially motility-enhancing prostaglandins. What about the prostate gland's contribution?
The prostate gland secretes a thin, milky fluid. Its key contents, as listed in the text, include calcium, citrate ion, phosphate ion, a specific clotting enzyme, and also a substance called profibrinolysin.
And its contribution to the overall volume.
The prostate adds about 30% of the total semen volume. Its muscular capsule also contracts simultaneously with the vas deferens during emission, expelling its fluid into the urethra to mix with the other components.
And you highlighted earlier the critical importance of the prostate's pH.
Absolutely crucial. The prostatic fluid is slightly alkaline. As we discussed, this alkalinity is necessary to neutralize the mildly acidic fluid coming from the vas deferens, and more importantly, the highly acidic environment of the vagina. Remember, sperm are most active at a pH between 6.0 and 6.5, and the prostatic fluid is key to achieving this favorable pH range for optimal motility and fertility.
And the milky appearance of semen comes primarily from the prostate fluid.
Yes, the text notes that the characteristic milky appearance is due to the prostatic fluid, while the more mucoid consistency comes from the seminal vesicles and the various mucus glands.
Okay, let's try to summarize the overall composition and properties of semen, putting it all together.
Right, so semen, as it's ejaculated, is this complex mixture of all these fluids. Roughly 10% is the fluid and sperm coming directly from the vas deferens. Almost 60% is the fluid from the seminal vesicles. Remember, added last to help flush the ducts. About 30% is the fluid from the prostate gland, and then small amounts of mucus are contributed by those urethral and bulbourethral glands.
So seminal vesicle fluid is the main component by volume, and the average pH is about 7.5 thanks to the prostate.
Correct. The alkaline prostatic fluid elevates the overall pH to about 7.5. This is slightly more alkaline than the absolute optimal 6.0, 6.5 range for peak sperm motility, but it's still far, far better than the damaging acidity of the vagina.
Tell me about that curious property the text mentions. Coagulation and then liquefaction, semen clots.
Yes, it's a really unique feature. The clotting enzyme that comes from the prostatic fluid interacts with the fibrinogen provided by the seminal vesicles. This interaction forms a weak coagulum, essentially a temporary gel or clot, shortly after ejaculation.
And what's the purpose of this temporary clot?
The text suggests this temporary coagulum serves to hold the semen in the deeper part of the vagina, near the cervix. This might prevent it from immediately leaking out and could help concentrate the sperm near the opening of the uterus, giving them a better starting point.
But it doesn't stay clotted for long, it dissolves.
No, exactly. Within about 15 to 30 minutes after ejaculation, this coagulum dissolves. This process is called liquefaction, and it's caused by an enzyme called fibrinolysin. Fibrinolysin itself is formed from profibrinolysin, which, interestingly, is also a component of the prostatic fluid.
So the prostate contributes both the clotting agent and the agent that dissolves the clot later. That's a remarkably sophisticated timed system.
It really is. And importantly, the text notes that sperm are relatively immobile immediately after ejaculation, likely because they're trapped in the viscosity of this coagulum. But as the coagulum dissolves and liquefies, the sperm gain motility and are freed up, ready to begin their journey up the female reproductive tract.
And just to reinforce the lifespan details one more time.
Okay, again. potentially many weeks in the suppressed state within the male ducts, but only a maximum life of about 24 to 48 hours in the female tract at body temperature.
Yeah.
But, as we mentioned, they can be preserved for years when frozen properly below negative 100 degrees Celsius.
So semen is this incredibly carefully engineered vehicle. It's got the right volume, the right pH, nutrients, even these temporary physical properties like clotting and liquefying, all designed to deliver viable motile sperm effectively into the female reproductive tract.
It's a very complex delivery system. Yeah, optimized for that crucial initial phase of the journey. But, and this is key, even after being delivered into the female tract, the sperm still aren't fully capable of fertilization right away. They need further preparation, changes that happen within the female tract.
And this is the process called capacitation you mentioned earlier.
Yes, capacitation. The text emphasizes this point. Sperm leaving the epididymis, while structurally mature and capable of motility, are actively prevented from fertilizing the ovum by various inhibitory factors they pick up from the epithelia lining the male ducts. Capacitation refers to the collection of changes they undergo in the female genital tract that remove these inhibitors and make them fully capable of fertilization.
How long does this capacitation process usually take?
The text states that it normally requires anywhere from 1 to 10 hours for capacitation to occur within the female tract, so it's not instantaneous.
What are the key changes that actually happen to the sperm during capacitation?
There are several important transformations that occur when sperm come into contact with the fluids in the female genital tract, specifically in the uterus and the fallopian tubes. First, those various inhibitory factors that suppress their activity and fertilizing capability back in the male ducts are effectively washed away by the female tract fluids.
Okay, so step one is removing those inhibitory brakes. What else happens?
Well, a major change involves the sperm membrane, specifically the membrane around the acrosome, that enzyme cap. While they're traveling through the male ducts, sperm are exposed to numerous cholesterol-containing vesicles secreted by the seminiferous tubules. This cholesterol deposits onto the acrosome membrane, making it tougher and more stable, which actually prevents a premature release of those potent acrosome enzymes.
Okay.
Then, in the female tract, the sperm swim away from these cholesterol vesicles, and over several hours, they gradually lose much of that excess cholesterol from their surface membrane.
So losing cholesterol actually weakens the acrosome membrane. That seems a bit counterintuitive.
It does, doesn't it? But this weakening is actually necessary. It prepares the membrane for the next critical step, the acrosome reaction. Another key change during capacitation is that the sperm membrane becomes much more permeable to calcium ions.
Calcium again. We saw its importance in the prostate fluid. What does calcium do when it gets into the sperm during capacitation?
This influx of calcium ions into the sperm has two major effects, according to the text. First, it dramatically changes the way the tail moves. It transforms the previous weak, undulating motion into a powerful, whip-like action, significantly increasing the sperm's motility and propelling it forward much more effectively. Think hyperactivation. Second, the calcium influx causes crucial changes in the membrane covering the acrosome itself, making it much easier for the acrosome to rapidly release its enzymes when the sperm finally contacts the ovum.
So capacitation basically makes them stronger, faster swimmers, and primes the enzymatic drill kit for action.
That's a great way to put it, exactly. The text makes it very clear. Without these capacitation changes, the sperm simply cannot penetrate the ovum to cause fertilization. It's an essential step.
All right, so once they're capacitated, they're finally ready for the target, the ovum. How do they use those primed acrosome enzymes to actually get inside?
Okay, so the text describes the ovum, the egg cell, as being surrounded by multiple layers of granulosa cells. These are cells that were expelled along with the ovum from the ovary during ovulation. Underneath these granulosa layers is a thick, protective outer layer called the zona pellucida. To achieve fertilization, the sperm must first digest its way through these granulosa cell layers and then penetrate the much tougher zona pellucida.
And that's where the acrosome enzymes are finally deployed.
Yes, the stored acrosomal enzymes, remember, the hyaluronidase and the proteolytic enzymes, begin to be released as the sperm encounters these outer layers. The hyaluronidase is particularly effective at breaking down the connections between the granulosa cells, creating pathways through these layers so the sperm can reach the surface of the zona pellucida itself.
What happens when a capacitated sperm actually reaches the zona pellucida?
This is a key moment. The anterior membrane of the sperm head contains specific receptor proteins that recognize and bind tightly to complementary receptor proteins located in the zona pellucida. This specific binding event acts as the trigger for what's called the acrosome reaction.
Describe the acrosome reaction. What happens then?
In the acrosome reaction, the entire acrosome, that enzyme-filled cap, rapidly dissolves or breaks down, releasing all of its stored enzymes at once, right onto the surface of the zona pellucida where the sperm is bound.
A full payload release.
Exactly. Within minutes, these enzymes, particularly the powerful proteolytic ones, digest a pathway through the thick zona pellucida, allowing the sperm head to pass through this barrier and reach the actual leucite membrane underneath.
So binding to the zona pellucida is the trigger for that final decisive enzymatic push.
It's the trigger for releasing the full enzymatic payload needed to breach that main protective barrier, the zona pellucida.
And once the sperm head gets through the zona and reaches the oocyte membrane,
Within about another 30 minutes, the cell membranes of the sperm head and the oocyte itself fuse together, becoming one continuous membrane, forming a single cell. Simultaneously, the genetic material, the chromosomes and genes from both the sperm and the oocyte, combine within this newly formed cell to create a completely new cell genome.
that's the moment.
That is the moment of fertilization. It results in a single cell, the zygote, that now contains an equal contribution of genetic material from both the mother and the father.
And at this point, the text notes, embryo development begins. That's just incredible. Such a complex journey of chemical and physical transformation, all leading to this single fusion event.
It truly is remarkable. But considering that hundreds of millions of sperm might be present in the vicinity, how does the system prevent more than one sperm from entering the ovum? Polyspermy, multiple fertilizations, would be genetically disastrous.
Right. The text describes a very rapid mechanism for this block, triggered by the very first successful penetration. Within just a few minutes after the first sperm penetrates the zona pellucida and fuses with the oocyte membrane, calcium ions begin to diffuse inward through the oocyte membrane into the oocyte cytoplasm.
Calcium again, playing yet another critical role in the process.
Yes, calcium seems to be a key signaling molecule throughout. This influx of calcium into the oocyte causes the oocyte itself to release the contents of multiple tiny sacs called cortical granules. These granules release their contents by exocytosis into the paravital lane space, that's the narrow space just outside the oocyte membrane but inside the zona pellucida. What do these cortical granules contain and what do they actually do once released?
The substances contained within these granules rapidly permeate throughout the entire substance of the zona pellucida. Their primary effect is to chemically alter the zona pellucida in such a way that it blocks the binding sites for any additional sperm trying to attach. The text also mentions that these substances cause any sperm that might have already begun binding to the zona pellucida to detach.
So the ovum actively seals itself off, chemically modifying its outer layer almost instantly after the first sperm gets through.
That's precisely what happens. It's a rapid defense mechanism, sometimes called the zona reaction or the cortical reaction. It's a crucial block to polyspermy, ensuring that almost always only a single sperm enters the oocyte, which is essential for viable embryo development.
That level of coordinated, rapid-fire biological response is just astounding. From a non-motile spermatid deep in the testes through maturation, capacitation, enzymatic penetration, culminating in the ovum's own sophisticated defense mechanism, it's a truly complex and fascinating biological narrative.
It really is. But like any complex biological system, it's unfortunately susceptible to issues and breakdowns. The text moves on to discuss various abnormalities of spermatogenesis and factors affecting male fertility.
What are some of the things that can damage the seminiferous epithelium, the actual sperm-producing tissue in the tubules?
The text lists several potential causes for destruction or damage to the seminiferous epithelium. These include certain diseases, like bilateral orchitis, that's inflammation of both tests, which can sometimes be a complication of mumps infection in adult males and can lead to permanent sterility. Congenital issues, things present at birth like strictures or blockages of the genital ducts or other developmental abnormalities, can also cause the tubular epithelia to degenerate. And finally, as we've touched upon, excessive temperature of the testes can lead to at least temporary sterility by causing degeneration of most of the seminiferous tubule cells, except, interestingly, the very initial spermatogonia.
Ah, temperature again. That really underscores why the testes are located outside the main body cavity in the scrotum.
Exactly. The source explicitly states that increasing the temperature of the testes, even by a few degrees, can prevent spermatogenesis. Maintaining testicular temperature several degrees below the internal body temperature is absolutely essential for normal sperm formation. The scrotum acts as this critical temperature regulator, keeping the testes normally about 2 degrees Celsius below the internal body temp.
And the scrotum even has reflexes to help maintain that optimal temperature.
Yes, it does. In cold conditions, scrotal reflexes cause the scrotal musculature to contract, pulling the testes closer up towards the warmth of the body to help maintain that crucial temperature differential. Conversely, in warm conditions, the muscles relax, allowing the testes to hang lower, away from the body, facilitating cooling. It's a clever built-in thermostat, essentially.
What about cryptorchidism? That's when a testes doesn't descend properly into the scrotum, right?
Correct. Cryptorchidism is the failure of one or both testes to descend from their original developmental position in the abdomen down into the scrotum, which should normally happen at or near the time of birth. Remember, testes normally develop up in the abdomen and then descend through the inguinal canals during the last 2 to 3 months of gestation. Cryptorchidism means this descent is incomplete or just doesn't happen, leaving the testes stuck in the abdomen, the inguinal canal, or somewhere else along the path.
And if a testes remains stuck up in the abdomen, can it produce sperm normally?
No. According to the text, an undescended testes that remains in the abdominal cavity throughout life is incapable of forming sperm. The higher temperature inside the abdomen causes the tubular epithelium, the sperm-producing cells, to degenerate over time, leaving only the interstitial structures, like the Leydig cells, which can still produce testosterone.
So is this primarily considered a temperature effect as well?
The text mentions that it's often claimed that the slightly higher temperature in the abdomen compared to the scrotum is sufficient by itself to cause this degeneration of the tubular epithelium and result in sterility. Although it does add a small note of caution that this point isn't absolutely certain based on the evidence presented.
Can cryptorchidism be treated to restore fertility?
Well, surgical operations can often be performed, ideally before puberty, to successfully relocate cryptorchid tests down into the scrotum. If done early enough, this can potentially allow normal spermatogenic function to develop and preserve fertility.
And what causes the failure of descent in the first place? Is it just mechanical?
Not always. The normal stimulus for testicular descent during fetal life is testosterone secretion by the fetal tests themselves. The text suggests that many cases of cryptorchidism are actually caused by abnormally formed tests that simply don't secrete sufficient testosterone during that critical fetal period. In these cases, even if surgery successfully relocates the testes to the scrotum, it might still not result in functional sperm production because the underlying issue is with the testis itself, not just its location.
So temperature sensitivity and proper anatomical location are clearly vital for spermatogenesis. Beyond those factors, the text discusses sperm count and sperm quality as major factors in male fertility.
Yes. A normal male ejaculate, the text states, is typically around 3.5 milliliters in volume and contains an average concentration of about 120 million sperm per milliliter.
That's a staggering number, a total of around 400 million sperm per ejaculation on average.
Yes, it's huge, although there's definitely a wide range, even among fertile men. The text mentions concentrations can vary anywhere between 35 million and 200 million per milliliter.
What count is generally considered too low for fertility? What's the threshold?
The text indicates that if the sperm count consistently falls below about 20 million per milliliter, the person is likely to be infertile.
That's a really huge difference between the average of 120 million and the infertility threshold of 20 million. Why so many needed?
It is a big difference. And the text actually points out this seeming paradox. Why are hundreds of millions of sperm apparently required for a single fertilization event to occur? It notes that the precise reasons for this massive numerical requirement aren't fully understood from the information available in the source material.
Okay, so count is definitely important. But what about the quality of the sperm? Can that be an issue too?
Absolutely. That's another critical factor. Even if a man has a sufficient sperm count, infertility can still occur if the sperm produced are morphologically abnormal, meaning they have defects in their shape or structure, or if they have poor motility, meaning they can't swim effectively.
What kind of morphological abnormalities are described?
The text references figure 81-5, which apparently shows examples like sperm with 2 heads, or abnormally shaped heads, or defective abnormal tails. If a significant proportion of the sperm produced have these kinds of structural defects, they are much less likely to be capable of successfully navigating the female tract and fertilizing the ovum.
And is it possible for sperm to look structurally normal but still be unable to swim properly?
Yes, the text notes that for reasons that aren't entirely understood, sperm can sometimes appear morphologically normal under a microscope, but still be non-motile or relatively non-motile. So if the majority of sperm in an ejaculate are either morphologically abnormal or lack adequate motility, the individual is likely to be infertile, regardless of their actual sperm count.
So male fertility is really this complex interplay of factors, needing a sufficient count of sperm, but also needing those sperm to have the proper morphology or structure and adequate motility or swimming ability. Issues with any one of those can lead to infertility.
It really highlights that just producing large numbers of cells isn't enough. They must also be properly formed and fully functional to do their job.
Okay, shifting gears now, let's explore the physiological events of the male sexual act itself. The text breaks this down starting with the neuronal stimuli involved.
Right. The primary driver of sexual sensation, according to the text, originates in the gland's penis. It contains a very sensitive system of specialized sensory end organs.
And how are these sensory organs typically stimulated?
The massaging action of intercourse on the glands is the main stimulus. This transmits what the text calls sexual sensation, signals along the pudendal nerve into the sacral plexus, then up to the sacral region of the spinal cord, and ultimately the signals reach the brain.
Are there other areas of the body that contribute to this sensation?
Yes. The text mentions that impulses contributing to sexual sensation also come from adjacent areas like the anal epithelium, the scrotum, and other perineal regions. It also notes that sexual sensations can actually originate from internal structures as well, like parts of the urethra, the bladder, the prostate, the seminal vesicles, the testes, and the vas deferens.
It even states that the simple filling of the sexual organs with secretions can contribute to sexual drive.
That's noted as a potential contributing cause, yes. The text also makes the interesting point that even mild infusion or inflammation in these pelvic organs can sometimes stimulate sexual desire. And it briefly touches on certain aphrodisiacs like cantharidin, which apparently work by irritating the mucous membranes, causing inflammation and vascular congestion, which can indirectly stimulate desire.
So a complex mix of external and internal physical stimuli. What about the psychological factors involved?
Psychic stimuli play a very significant role. The text notes that appropriate psychic factors, thoughts, emotions, visual stimuli can greatly enhance the ability to perform the sexual act. In fact, simply thinking sexual thoughts or even dreaming about sexual activity can sometimes be enough to initiate the physiological process, potentially leading all the way to ejaculation. The text gives nocturnal emissions, or wet dreams, as a common example of this, especially in teenagers.
Does the brain absolutely have to be involved for the act, particularly ejaculation, to occur, or is it more reflexive?
Interestingly, the text suggests it's not strictly necessary, at least for the basic mechanics. While psychic input from the brain is obviously hugely important for desire and performance, there are inherent reflex mechanisms integrated within the sacral and lumbar regions of the spinal cord. The text states that these spinal reflexes can, in some cases, cause ejaculation even after the spinal cord has been transected above the lumbar region, cutting off direct input from the brain. So the basic reflex arc resides in the lower cord and can be triggered either by psychic stimulation routed down from the brain or by direct physical stimulation of the sex organs, or, most commonly, a combination of both.
Fascinating. Let's detail the actual physiological stages of the male sexual act as described in the text.
Okay. The first and often most recognized effect is, of course, penile erection.
How is erection controlled neurologically?
Erection is primarily mediated by parasympathetic nerve impulses. These originate in the sacral segments of the spinal cord and travel via the pelvic nerves to the penis. Now, a key detail the text provides is that these particular parasympathetic fibers release nitric oxide, NO, and vasoactive intestinal peptide, VIP, in addition to the usual parasympathetic neurotransmitter, acetylcholine. This is somewhat unusual.
And how do NO and VIP actually cause the erection? What's the mechanism?
Nitric oxide seems particularly important here. It activates an enzyme called guanylyl cyclase within the smooth muscle cells. This enzyme then increases the intracellular concentration of cyclic GMP, or cGMP. This cGMP molecule is the key signal that causes relaxation of the smooth muscle in the walls of the arteries supplying the penis, and also relaxation of the smooth muscle within the trabecular meshwork of the erectile tissue itself, the corpus cavernosa and the corpus spongiosum, which are shown in figure 81-6.
So smooth muscle relaxation leads to increased blood flow into the erectile tissue.
Exactly. As this smooth muscle relaxes, there's a rapid and massive inflow of arterial blood under pressure into the large, normally relatively empty, cavernous sinusoids within the erectile tissue. This dramatically increased inflow combined with the simultaneous partial occlusion or compression of the veins draining the penis, causes these sinusoids to dilate immensely and fill with blood.
And that makes the penis rigid.
Right. The strong, relatively inelastic fibrous outer coats surrounding the erectile bodies, especially the corpora cavernosa, mean that as the sinusoids fill under pressure, the tissue balloons outwards against these coats, causing the penis to become hard, elongated, and erect.
That's a very precise description of the vascular hydraulics involved. What about lubrication during the act? Is that controlled differently?
Lubrication is also primarily a parasympathetic function. The same parasympathetic impulses that trigger erection also stimulate the urethral glands lining the urethra and those bulbourethral Cowper's glands near the base to secrete mucus. This mucus flows into and through the urethra, providing lubrication to aid intercourse.
The text notes that most of the lubrication during intercourse actually comes from the female organs, but the male contribution is still important.
Yes, it emphasizes that point. While the female lubrication is typically greater, some male contribution is necessary. Without satisfactory lubrication from either source, the male sexual act is seldom successful because painful grating sensations can occur, which severely inhibit sexual excitement and pleasure.
Okay, so parasympathetic nerves drive erection and lubrication. What about the climax, the processes of emission and ejaculation?
Emission and ejaculation are controlled by the sympathetic nervous system. As the level of sexual stimulus becomes intense, reflex centers located in the thoracolumbar spinal cord, specifically segments T12 to L2, begin sending sympathetic impulses out via the hypogastric and pelvic sympathetic nerve plexuses to the genital organs. These signals initiate the process of emission.
What exactly happens during emission? What defines that phase?
Emission is essentially the process of loading the semen into the internal urethra, preparing for expulsion. It begins with rhythmic contractions of the vas deferens and its ampulla, which expels sperm into the internal urethra near the prostate. This is quickly followed by contraction of the muscular coats surrounding the prostate gland, adding prostatic fluid to the mix. Then, the seminal vesicles contract, adding their fluid containing fructose and prostaglandins. All these fluids mix together in the internal urethra with the mucus already secreted by the bulbourethral glands to form the complete fluid we call semen. That collection process is emission.
And ejaculation immediately follows emission.
Yes. The filling of the internal urethra with this newly formed semen triggers sensory signals. These signals travel via the pudendal nerves back to the sacral spinal cord. The text says this arrival of semen in the urethra gives the subjective feeling of sudden fullness in the internal genital organs.
And these sensory signals trigger the expulsion.
Exactly. These sensory signals further excite and intensify the rhythmic contractions of the internal genital organs that began during emission. Crucially, they also cause strong rhythmic contractions of the skeletal muscles at the base of the penis, specifically the ischiocavernosus and bulbocavernosus muscles, which compress the bases of the penile erectile tissue.
Okay.
The combined effect of these ongoing internal organ contractions and the powerful external muscle contractions creates significant rhythmic pressure increases within the erectile tissue, the genital ducts, and the urethra. This pressure forcibly expels, or ejaculates, the semen from the urethra out to the exterior in spurts.
The text also mentions pelvic thrusting movements being part of this climax phase.
Yes, it notes that rhythmic contractions of various pelvic and some trunk muscles occur simultaneously, causing the characteristic thrusting movements of intercourse. These are believed to help propel the ejaculated semen deeper into the vagina and towards the cervix, maximizing the chances of sperm reaching the uterus.
So emission is the internal collection and mixing of semen components, and ejaculation is the forceful expulsion driven by sympathetic nerve signals culminating in coordinated internal and external muscle contractions.
Correct. And the text refers to the entire period covering emission and ejaculation collectively as the male orgasm. Following orgasm, sexual excitement typically disappears quite rapidly, usually within a minute or two, and the erection subsides. This return to the non-aroused state is known as the resolution phase.
That's a very clear step-by-step breakdown of the nervous system control and the precise physical mechanics of the male sexual act. From the initial sensory input, through the distinct roles of parasympathetic and sympathetic systems, to the specific muscle actions driving the different phases.
It really shows a complex sequence of coordinated events that culminates in the delivery of semen. Now let's delve into the hormones that regulate so much of what we've just discussed, focusing primarily on testosterone and the other male sex hormones, the androgens.
Right, the primary category is androgens, isn't it? What defines them?
Yes, androgens are basically defined as steroid hormones, characterized by their masculinizing effects. The main ones secreted by the testes are testosterone, dihydrotestosterone, often abbreviated DHT, and androstenedione. The text notes that testosterone is by far the most abundant one secreted by the testes, but a significant amount of it is actually converted in the target tissues into dihydrotestosterone, which in many cases is the more biologically active form of the hormone.
Where exactly in the testes is testosterone produced? Which cells?
It's produced by the interstitial cells of Leydig. These specialized cells are located in the spaces between the seminiferous tubules. You can see them in figure 81-7. They constitute about 20% of the total mass of the adult testes.
Do the Leydig cells have a consistent presence and activity level throughout a male's life?
They have a really fascinating pattern of activity. They are almost completely absent during childhood, between infancy and puberty. However, they are numerous and active in newborn male infants for the first few months of life, and then they reappear and become numerous and highly active again in adult males after puberty kicks in. Both of these periods, early infancy and post-puberty adulthood, correspond to times of significant testosterone secretion. The text even points out that tumors originating from Leydig cells can secrete very large quantities of testosterone.
And if the seminiferous tubules, where the sperm are actually made, get damaged, do the Leydig cells typically survive and keep making testosterone?
Often, yes. The text mentions that if the germinal epithelium lining the tubules is destroyed by agents like excessive heat or radiation, X-rays, the Leydig cells are usually less susceptible to that damage and can often continue to produce and secrete testosterone normally.
Are androgens produced anywhere else in the body besides the testes?
Yes, the adrenal glands, located on top of the kidneys, also secrete at least 5 different androgens. However, their total masculinizing activity in a normal adult male is typically quite low, usually less than 5% of the total contribution from the testes. These adrenal androgens primarily contribute to the development of pubic and axillary armpit hair in both sexes.
But the text mentions that adrenal tumors can sometimes cause significant masculinization.
Yes, that's an important exception. If a tumor develops in the adrenal cortex of the cells that produce androgens, the amount of androgenic hormone secreted can become very high enough to cause significant masculine characteristics to develop, even in a female. This is discussed elsewhere in physiology texts in relation to adrenogenital syndrome.
And are androgens present in females at all, normally?
The normal ovary produces only minute physiologically insignificant quantities of androgens. However, the text notes that rarely, specific types of ovarian tumors called arenoblastomas, which originate from embryonic rest cells within the ovary, can secrete excessive quantities of androgens, leading to masculinization in women.
Chemically speaking, these are all steroid compounds derived from cholesterol.
Yes, the text confirms they are all steroid hormones, structurally similar to cholesterol, from which they are synthesized in both the testes and the adrenal glands. Figure 81-8 shows the chemical structures of testosterone and dihydrotestosterone.
Once testosterone is secreted by the Leydig cells into the bloodstream, how does it travel around the body? Is it free or bound?
Most of it travels bound to proteins. About 97% of the secreted testosterone becomes bound to plasma proteins. Some binds loosely to albumin, but much of it binds more tightly to a specific carrier protein called sex hormone binding globulin, SHBG, which is a type of beta globulin. It circulates in this bound state for anywhere from 30 minutes to several hours before it's either taken up by tissues or degraded into inactive forms for excretion.
And you mentioned earlier that conversion to dihydrotestosterone happens in the target tissues.
Yes, this is a really significant metabolic step for testosterone's action in certain places. Within many target cells particularly, the text highlights, in the prostate gland and in the external genitalia of the developing male fetus, there's an enzyme called 5-reductase. This enzyme converts testosterone into dihydrotestosterone, DHT. Many of testosterone's effects, especially those promoting growth and function in these specific organs, are actually mediated by DHT after this local conversion.
How is testosterone eventually broken down and removed from the body when it's done its job?
Testosterone that doesn't bind to tissues and exert an effect is rapidly metabolized and inactivated, primarily in the liver. The liver converts it into inactive products like androsterone and dehydroepiandrosterone. These metabolites are then chemically conjugated, typically with glucuronides or sulfates, which makes them more water-soluble and easier for the body to excrete. These conjugated inactive products are then excreted either into the gut via the liver's bile production or, more significantly, by the kidneys into the urine.
What about estrogen production in males? The text mentions this too, which might seem counterintuitive to some.
It might seem that way, but yes, males do produce a small but physiologically measurable amount of estrogens. The text estimates it's about one-fifth the quantity found in a non-pregnant female. These estrogens can be recovered and measured in male urine.
Where does this estrogen come from in males? What are the sources?
The text points to a couple of important sources. First, there's actually a high concentration of estrogens found within the fluid inside the seminiferous tubules themselves. It's thought that this locally produced estrogen might be important for the process of spermatogenesis, that final sperm differentiation stage. This testicular estrogen is believed to be formed by the Sertoli cells, which apparently have the enzymatic machinery, aromatase, to convert some of the testosterone produced by the nearby Leydig cells into estradiol, a potent estrogen.
Okay, so some is made right in the testes. Is that the main source?
No, actually. The text states that a much larger quantity of estrogen, accounting for about 80% of the total male estrogen production, is formed from the conversion of testosterone and another androgen, androstenediol, in various other tissues throughout the body outside the testes. The liver is mentioned as a particularly important site for this peripheral conversion.
So testosterone is the main male hormone secreted by the testes, but it gets converted to the more potent DHT in some key target tissues, and it also serves as a precursor for estrogen production, both locally in the testes by Sertoli cells and more significantly in peripheral tissues like the liver. That's a complex metabolic picture for sure.
It really shows the interconnectedness of steroid hormone synthesis and metabolism. Now let's shift focus to the major functions of testosterone itself, what it actually does in the body throughout life.
The text broadly states it's responsible for the distinguishing masculine body characteristics. That's the big picture.
That's the overarching role, yes. And its influence begins incredibly early in development, starting around the 7th week of embryonic life. The fetal testes, stimulated initially by human chorionic gonadotropin, HCG, coming from the placenta, begin secreting testosterone. This secretion is crucial and continues throughout fetal development, and even for about 10 weeks or so after birth, before tapering off dramatically until puberty hits.
And this early fetal testosterone is absolutely key for establishing the male anatomical blueprint, right? Overriding the female default.
Absolutely critical. The text explains the fundamental genetic switch. The SRY gene, located on the male Y chromosome, produces a protein called SRY protein, or testes determining factor. This SRY protein initiates a cascade of events that causes the cells of the embryonic genital ridge to differentiate into testes, which then start secreting testosterone. Without the Y chromosome and the SRY gene, the genital ridge follows the default pathway and develops into estrogen-secreting structures, the ovaries. So the hormone environment created by the testes is key.
And the text makes this point quite powerfully, with examples from experiments where hormone exposure can actually override genetic sex development.
Yes, it cites classic experimental evidence. Injecting male sex hormone, testosterone, into pregnant animals can lead to the development of male sexual organs, even in genetically female X fetuses. Conversely, removing the testes from a male XY fetus very early in development results in the development of female sexual organs instead. This really demonstrates the powerful directive influence of the hormonal environment, specifically testosterone, on phenotypic sex development.
So fetal testosterone literally builds the male reproductive tract and the external genitalia?
Pretty much. It's directly responsible for the development of the penis and scrotum instead of the clitoris and vagina, and internally for the formation of the prostate gland, the seminal vesicles, and the male genital ducts like the epididymis and vas deferens. And importantly, at the same time it's promoting male development, it actively suppresses the formation of female internal organs from structures that would otherwise develop.
And it also plays a role in stimulating the descent of the testes into the scrotum.
Yes, that's another critical function of fetal testosterone. It provides the hormonal stimulus for the testes to descend from the abdomen down into the scrotum, a process that normally occurs during the last 2 to 3 months of gestation. The text notes that administering testosterone or gonadotropic hormones, which stimulate the Leydig cells to produce testosterone, can sometimes be used clinically to induce descent in male children born with undescended testes, cryptorchidism, provided the inguinal canals are anatomically open enough to allow passage.
Okay, so testosterone shapes the male body plan during embryonic and fetal development. What about the dramatic physical changes that occur at puberty?
Right, the significant increase in testosterone secretion that happens at puberty, driven by the rising levels of gonadotropic hormones, LH and FSH, from the pituitary, as clearly illustrated in figure 81-9, which shows the surge in plasma testosterone levels is what causes the development of the adult primary and secondary sexual characteristics.
Primary characteristics meaning the sexual organs themselves enlarge and mature.
Yes, exactly. The penis, scrotum, and testes all increase in size quite dramatically, roughly eightfold between childhood and full maturity around age 20.
And the secondary sexual characteristics are all the other physical traits that typically distinguish adult males from adult females.
Correct. These are the characteristics that develop under testosterone's influence after puberty begins. The text lists several key ones. Body hair distribution. Testosterone causes the growth of hair in specific patterns on the pubis, often extending up the midline of the abdomen, the linea alba, sometimes to the umbilicus, or even higher, on the face, beard and mustache, on the chest, and sometimes on the back. It also generally makes hair on other parts of the body, like limbs, more prolific than in females. Male pattern baldness. This is an interesting one. The text clarifies that typical male pattern baldness requires two factors working together. First, a genetic predisposition, and second, the presence of large quantities of androgenic hormones like testosterone and DHT. A man without functional tests doesn't typically become bald in this pattern. Conversely, many genetically predisposed men who are virile, producing plenty of androgens, will experience hair loss on the top of the head. It also notes that a woman who has the genetic background for baldness and then develops an androgen-secreting tumor can develop baldness just like a man's voice. Testosterone causes hypertrophy, or enlargement, of the laryngeal mucosa, the lining of the voice box, and enlargement of the larynx itself. This leads to the breaking and subsequent deepening of the voice from the higher pitch of childhood to the typical adult masculine voice. Skin thickness and acne. Testosterone increases the thickness and overall ruggedness of the skin across the body. It also significantly increases the rate of secretion from the sebaceous glands, especially those on the face. This increased sebum production is often the cause of acne, which is described as one of the most common features of male adolescence when the skin is first exposed to significantly increased testosterone levels. The text notes the skin typically adapts to this hormonal influence after several years.
The text calls testosterone's effect on protein formation and muscle development arguably its single most important masculine characteristic.
That's a really key point, yeah. Testosterone is a potent anabolic steroid, meaning it promotes tissue building, especially protein synthesis. After puberty, males typically experience roughly a 50% greater increase in muscle mass compared to females, and this is largely due to testosterone's powerful effect on increasing the rate of protein synthesis not just in muscles, but throughout the body. This generalized protein building effect also contributes to some of the other changes, like those in the skin and the voice box.
And this anabolic effect is why synthetic androgens are sometimes misused by athletes, but also why they carry significant health risks.
Precisely. The text explicitly mentions this misuse by athletes and strongly deprecates it due to the potentially severe harmful side effects, which it says are discussed in more detail elsewhere. It also notes the sometimes questionable use of testosterone as a youth hormone in older age to combat frailty, and importantly cautions about recent studies suggesting a possible increased risk of cardiovascular events associated with testosterone replacement therapy in some groups of older men.
Beyond muscle, does testosterone have significant effects on bones as well?
Yes, very significant effects. After puberty, under the influence of testosterone, bones become considerably thicker and deposit more calcium salts, making them denser and stronger. Testosterone increases the total amount of bone matrix, the protein framework, and also promotes calcium retention by the body. This effect on bone is believed to be linked both to its general protein anabolic function, building the matrix, and potentially a direct effect stimulating calcium deposition in response to that matrix.
Does it affect the shape of bones, like the pelvis?
Yes, it has quite specific effects on the pelvis. Testosterone causes the male pelvis to develop its characteristic shape, relatively narrow outlet, longer overall, more funnel shaped compared to the broader, ovoid female pelvis. These changes greatly increase the strength of the male pelvis for load bearing. The text notes that without testosterone during development, the male pelvis would tend to develop more along the female pattern. Because of its ability to increase bone size and strength, testosterone is sometimes used therapeutically to treat osteoporosis in older men.
What about height? Does testosterone make men taller? It increases bone growth, right?
This has a somewhat nuanced answer according to the text. If there are abnormally high levels of testosterone present in a growing child, for example due to a tumor, it causes a marked increase in the rate of bone growth and a significant spurt in height. But the ET, and this is the crucial part, it also causes the epiphyses, the growth plates at the ends of the long bones, to fuse with the shafts of the bones at an earlier age than they normally would.
Ah, so it stops growth sooner.
Exactly. This early closure of the growth plates ultimately prevents the individual from reaching the full potential height they might otherwise have achieved. The text even makes the interesting point that normal men who experience the typical pubertal testosterone surge are usually slightly shorter on average than individuals who are castrated before puberty. Eunuchs. Eunuchs lack the pubertal growth spurt driven by testosterone, but their epiphyses close much later, allowing their bones to continue growing for a longer period.
That's a surprising trade-off. A faster growth spurt initially, but an earlier end to overall growth, resulting in slightly less final height compared to someone without the pubertal testosterone surge.
It really illustrates the complex hormonal balance involved in regulating growth. What about basal metabolic rate? BMR. Does testosterone affect that?
The text says that large quantities of testosterone can increase BMR by as much as 15%. Even the usual amount present during adolescence and early adult life typically increases BMR by about 5 to 10% above the value it would be without active tests. This effect is possibly indirect, perhaps resulting from the overall increase in protein anabolism, more proteins, including enzymes, leading to increased overall cellular activity.
And its effect on red blood cell count is also noted.
Yes, apparently injecting normal physiological quantities of testosterone into a castrated adult results in a noticeable 15 to 20% increase in the number of red blood cells per cubic millimeter of blood. On average, the text states, men have about 700,000 more red blood cells per cubic millimeter than women.
Is the mechanism clear?
Interestingly, the text points out a bit of a contradiction here. While there's a strong association, it says testosterone doesn't seem to directly increase the production of erythropoietin, the main hormone stimulating red blood cell production, or directly stimulate red blood cell production in the bone marrow. So it suggests the effect might be indirect, perhaps linked again to the increased overall metabolic rates, somehow stimulating red blood cell production secondarily.
Okay. Finally, the text mentions a minor note on electrolyte and water balance.
Right. It mentions that testosterone has a minor effect on increasing the reabsorption of sodium by the kidneys, similar to, but much less potent than the adrenal mineralocorticoids like aldosterone. However, it notes that after puberty, the total blood volume and extracellular fluid volume in males does increase by about 5 to 10% relative to body weight, which might be related to this slight sodium retention or perhaps other metabolic effects. So, putting it all together, testosterone is this incredibly potent hormone. It influences everything from the very blueprint of sex determination in the fetus to shaping muscle mass, bone density, and shape, hair growth patterns, voice pitch, skin characteristics, basal metabolism, and even red blood cell count and fluid balance. It really sculpts many of the physical characteristics we typically recognize as masculine throughout life.
It's a profound influencer on development and ongoing physiology, absolutely. But how does it actually achieve these widespread and diverse effects down at the cellular level? The text offers some insight into its basic intracellular mechanism of action.
How does testosterone signal cells to, for example, build more protein?
The text explains that most of testosterone's diverse effects ultimately result from its ability to increase the rate of protein formation, or synthesis, within its target cells. It uses the prostate cell as a prime example. When testosterone enters a prostate cell, which happens within minutes after secretion, it's often converted, right there inside the cell, into dihydrotestosterone, DHT, by that enzyme 5-reductase we mentioned.
So DHT is often the active player inside the cell, not testosterone itself.
Yes, particularly in organs like the prostate, DHT seems to be the primary active form. This DHT molecule then binds to a specific receptor protein located in the cytoplasm of the cell. This newly formed hormone receptor complex then migrates together into the cell nucleus.
And what happens once it's inside the nucleus?
In the nucleus, the DHT receptor complex binds to another specific nuclear protein, likely associated with the DNA. This binding event acts like a switch, inducing the process of DNA-RNA transcription for specific genes. Within about 30 minutes, the activity of RNA polymerase, the enzyme that makes RNA copies from DNA, increases, leading to a progressive increase in the concentration of various types of RNA molecules within the cell.
And more RNA translates into more protein synthesis.
Exactly. This increase in RNA molecules provides the templates and machinery needed to drive a progressive increase in the synthesis of specific cellular proteins. The text notes that over a period of several days, this process can even lead to an increase in the quantity of DNA itself, indicating an increase in the number of prostatic cells through cell division. So testosterone, or DHT, stimulates both protein synthesis within existing cells and potentially cell proliferation in target tissues like the prostate.
This general mechanism, a steroid hormone entering a cell, binding to a receptor, the complex moving to the nucleus, and then altering gene expression to change protein production is a common theme for steroid hormones, isn't it? But seeing it described specifically for testosterone helps ground how it achieves its widespread effects.
It really explains how it can act as a master switch, turning on the production of specific proteins that are characteristic of its target organs, leading to those primary and secondary male sexual characteristics. The text does briefly mention, as an aside, that some studies suggest testosterone might also have rapid, non-genomic effects that don't require changes in gene expression or protein synthesis, but it notes that the physiological role of these rapid effects, if any, is yet to be determined based on this source material.
Okay, so the primary established mechanism is this alteration of gene expression leading to increased protein production. Moving on from the hormone itself and its actions, how is the production of these crucial hormones, and thus the regulation of overall male sexual function, controlled? We got a glimpse of the pituitary gland's involvement earlier.
Right. The whole control system is orchestrated by the brain, forming a classic endocrine feedback loop known as the hypothalamic pituitary testicular HPT axis. Figure 8110 in the text provides a nice illustration of this axis and its feedback controls.
It all starts in the hypothalamus, deep in the brain.
Yes, the starting signal comes from the hypothalamus, specifically from the secretion of gonadotropin-releasing hormone, or GnRH. This is a small peptide hormone, just 10 amino acids long, produced by specialized neurons located in the arcuate nuclei of the hypothalamus.
Where does the GnRH go once it's released by these neurons?
The nerve terminals of these GnRH-producing neurons end in a region at the base of the brain called the median eminence. Here, they release GnRH directly into a specialized capillary network called the hypothalamic hypophyseal portal vascular system. This unique portal blood system provides a direct route, carrying the GnRH from the hypothalamus down the pituitary stalk to the anterior pituitary gland.
And what does GnRH do when it reaches the anterior pituitary?
GnRH acts as the primary stimulus, signaling the specialized cells in the anterior pituitary, called gonadotropes, to synthesize and secrete the two key gonadotropic hormones that act on the testes, luteinizing hormone, LH, and follicle-stimulating hormone, FSH. The text notes that both LH and FSH are glycoproteins and are actually secreted by the same gonadotrope cells in the anterior pituitary. Importantly, the secretion of both LH and FSH almost completely stops if the stimulating signal from hypothalamic GnRH is absent.
Is GnRH released from the hypothalamus constantly, or does it come in bursts?
This is a really important point for how the system works. GnRH is secreted intermittently in distinct pulses. These pulses typically last only a few minutes at a time, but occur rhythmically, usually every 1 to 3 hours. The intensity of the signal reaching the pituitary is determined both by the frequency of these pulses and by the amount of GnRH released in each pulse.
How does the pituitary gland respond to these pulses of GnRH? Does it release LH and FSH in pulses too?
LH secretion from the anterior pituitary follows the pulsatile release of GnRH quite faithfully, meaning LH is also secreted in distinct pulses. As we'll see, LH is the primary stimulus for testosterone secretion by the Leydig cells in the testes. In fact, the amount of testosterone produced by the testes is directly proportional to the amount of LH available. FSH secretion, however, behaves a bit differently. Its level changes much more slowly, often over hours, in response to longer-term changes in the overall GnRH signal, and it only fluctuates slightly with each individual GnRH pulse.
So, LH release is pulsatile and closely linked to driving testosterone production, while FSH release is more steady and linked to its own specific function.
Correct. FSH's primary role, as mentioned earlier, is to stimulate the process of spermatogenesis. It does this by binding to specific FSH receptors located on the surface of the Sertoli cells within the seminiferous tubules. This binding causes the Sertoli cells to grow and to secrete various substances that are essential for nurturing and supporting the different stages of sperm development. The text clarifies, however, that both FSH and testosterone, which diffuses over from the nearby Leydig cells under LH stimulation, are necessary to properly initiate and maintain spermatogenesis.
Okay, so LH stimulates the Leydig cells to make testosterone, and FSH stimulates the Sertoli cells to support sperm development. But that support function also requires the presence of testosterone. It's a coordinated effort.
That's the functional division, yes. Now, how do the testes signal back to the brain and pituitary to regulate this whole system? That's where the crucial negative feedback loops come in.
How does testosterone itself provide feedback to control its own production?
Testosterone, secreted by the testes in response to LH stimulation, exerts a negative feedback effect, primarily inhibiting the anterior pituitary secretion of LH. The text explains that this inhibition happens mostly through a direct effect of testosterone acting back on the hypothalamus, where it decreases the secretion of GnRH. Less GnRH, released from the hypothalamus, means less stimulation of the anterior pituitary, leading to decreased secretion of both LH and FSH. The decreased LH then leads to reduced stimulation of the Leydig cells and consequently reduced testicular testosterone secretion.
So if testosterone levels get too high, it effectively tells the brain to slow down the signals, which then brings testosterone levels back down toward the normal range?
Exactly. It's a classic self-regulating negative feedback loop, automatically adjusting testosterone production to maintain relatively stable levels. Conversely, if testosterone levels fall too low, the negative feedback is reduced, leading to increased GnRH, increased LH and FSH, and therefore increased stimulation of the testes to produce more testosterone.
Is there a separate feedback mechanism related specifically to the process of sperm production itself, beyond just the testosterone level?
Yes, there is. This involves a different hormone called inhibin, which is secreted by the Sertoli cells, the same cells that support spermatogenesis under FSH stimulation. The text notes an important observation. When the semenstrous tubules are failing to produce sperm effectively, for instance if they're damaged, the secretion of SSH from the anterior pituitary increases markedly. Conversely, if spermatogenesis is proceeding too rapidly, the pituitary secretion of FSH tends to diminish.
And inhibin is the messenger carrying that signal back from the Sertoli cells.
Exactly. Inhibin, which has been isolated and identified as a glycoprotein produced by cultured Sertoli cells, provides this specific negative feedback signal. It acts directly on the anterior pituitary gland, selectively inhibiting the secretion of FSH with less effect on LH. The text emphasizes that this inhibin feedback system, which helps regulate the rate of spermatogenesis, operates simultaneously and in parallel with the testosterone feedback loop that primarily controls testosterone secretion.
So the system has these two distinct arms of negative feedback originating from the testes. Testosterone feeding back mainly on GnRH/LH release to control androgen levels, and inhibin from the Sertoli cells feeding back on FSH release to modulate the rate of spermatogenesis.
It's a sophisticated dual control feedback system, ensuring that both the hormonal output, testosterone, and the gamete production, spermatogenesis, are regulated appropriately based on the body's needs and the testes' functional state. The text also briefly brings up the role of human chorionic gonadotropin, HCG, again here.
Right. We saw that HCG from the placenta stimulates the fetal test early in development. Where else does it play a role?
Well, its primary role is during pregnancy. It's secreted in large amounts by the placenta and circulates in both the mother and the fetus.
And its effect on the sexual organs is similar to which pituitary hormone, LH or FSH?
The text states clearly that HCG has almost the same effects on the sexual organs as LH. Critically, in a developing male fetus, this placental HCG provides the essential stimulus for the fetal testes to secrete the testosterone needed to promote the formation and differentiation of all the male sexual organs. So it effectively acts as the initial LH signal, driving male development during gestation before the fetus's own pituitary testicular axis is fully functional.
Now, the text touches on the onset of puberty, noting it was a bit of a mystery for a long time. What's the current understanding of how puberty is triggered?
It was indeed a puzzle. The key understanding presented now is that during childhood, the hypothalamus secretes only very insignificant amounts of GnRH. Furthermore, the hypothalamus during childhood is extremely sensitive to the inhibitory feedback effects of any sex steroids, like the tiny amounts produced by the adrenals or testes. Even the slightest hint of sex steroid secretion has a very strong inhibitory effect on hypothalamic GnRH release, effectively keeping the whole system suppressed.
So the reproductive axis is actively held in check, suppressed throughout childhood.
Yes, exactly. Then, at the time of puberty, for reasons that the text admits are still not fully understood, the hypothalamic GnRH secretion somehow breaks through this profound childhood inhibition. The sensitivity to negative feedback decreases, and the hypothalamus starts releasing GnRH in those characteristic adult pulses. This increase in pulsatile GnRH secretion is the primary trigger that initiates the increased LH and FSH secretion from the pituitary, which in turn leads to the surge in testosterone production by the testes and the onset of adult sexual life with all its associated physical changes.
So it's like a brake is released on the hypothalamus at puberty, allowing the whole HPT axis to ramp up to adult levels. What about adult sexual life and the concept of the male climacteric or male menopause?
After puberty is initiated and adult function is established, gonadotropic hormones continue to be produced by the pituitary for the remainder of life, and some degree of spermatogenesis typically continues, potentially until death. However, the text does note that many men experience a slow, gradual decrease in their overall sexual functions, often starting sometime in their late 50s or 60s.
Is this decline inevitable or universal?
Not necessarily. The text highlights that there's considerable individual variation. It notes that many healthy men can remain virile and sexually active well into their 80s and even 90s. The decline, when it occurs, is often associated with other health factors like smoking, obesity, and underlying cardiovascular and metabolic issues such as hypertension, atherosclerosis, and type 2 diabetes, which can impair vascular and nerve function.
And is this age-related decline always directly due to falling testosterone levels?
The decline is related, at least in part, to a decrease in testosterone secretion by the aging testes. The general trend is shown in figure 81-9, with plasma testosterone levels tending to decrease somewhat with age. However, the text includes an important caveat, stating that some studies show plasma testosterone levels remain well within the normal range for many healthy older males. It quotes figures suggesting that perhaps only about 10-20% of men over 60, and maybe up to 50% of men over 80, are found to have plasma testosterone levels considered clinically low, for example, below 3 ng/ml or 300 ng/ml.
So low testosterone becomes more common in older men, but it's not the sole cause of age-related sexual decline, and it's certainly not experienced by all aging men.
That seems to be the conclusion from this test, yes. And it also mentions a clear association between the development of late-onset low testosterone and the presence of metabolic disorders like obesity and type 2 diabetes.
Yeah.
The relationship might be complex and bidirectional. This slow, gradual decline in male sexual function that can occur with aging is sometimes termed the male climacteric.
It's really fascinating to map out that entire hormonal axis from the pulsatile GnRH signal originating in the hypothalamus down to the pituitary releasing LH and FSH, stimulating the testes to produce testosterone and sperm, and then the intricate negative feedback loops involving both testosterone and inhibin regulating the whole system, and how this axis changes dynamically across the entire lifespan from fetal development through puberty, adulthood, and into older age.
It's a truly core regulatory system, orchestrating so much of male reproductive physiology. And of course, as with any complex biological system, there are potential abnormalities and dysfunctions that can occur. The text outlines several important ones, including issues related specifically to the prostate gland, conditions of hypogonadism, underfunction, hypergonadism, overfunction, usually due to tumors, and the common issue of erectile dysfunction.
Let's start with the prostate gland and the problems that are often seen, particularly with age.
Right. The prostate gland, as we know, is stimulated by testosterone to grow significantly at puberty. It typically reaches its adult size by around age 20 and remains relatively stable for decades. It might even involute or shrink slightly after age 50 if testosterone levels decrease significantly. However, a very common problem encountered in older men is a condition called benign prostatic fibroadenoma, often referred to as benign prostatic hyperplasia or BPH.
What exactly is that? Is it related to testosterone?
It's an abnormal, non-cancerous overgrowth of the prostate tissue itself. Importantly, the text clarifies that this benign hypertrophy is not directly caused by testosterone, even though testosterone was responsible for the initial growth of the gland at puberty. The exact cause is complex and multifactorial. This overgrowth, however, frequently causes significant urinary obstruction because the enlarged prostate squeezes the urethra that passes through it.
And prostate cancer is a completely different issue, right?
Yes, prostate cancer is a distinct pathological process and, as the text notes, a significant cause of male mortality, accounting for perhaps 2–3% of all male deaths. Unlike the benign overgrowth, the growth of cancerous prostate cells is often stimulated and accelerated by the presence of testosterone.
So treatment for prostate cancer often involves reducing or blocking testosterone's effects.
Precisely. Because testosterone can fuel the growth of many prostate cancers, a cornerstone of treatment, especially for advanced or metastasized cancer, often involves reducing testosterone levels or blocking its action. This can be achieved surgically by removing the testes, orchiectomy, or medically by administering drugs that suppress testosterone production, like GnRH analogs, or by giving estrogens, which inhibit pituitary LH secretion and thus reduce testicular testosterone production. These approaches can effectively slow the growth of the cancer, reduce bone pain from metastases, and prolong life, although they are typically not curative for advanced disease.
Okay, let's look at hypogonadism next. That's deficient function of the testes, right?
Yes, hypogonadism means the testes aren't functioning properly either in terms of hormone production or sperm production, or both. The consequences depend heavily on when this deficiency occurs during life.
What happens if the testes are non-functional during fetal life?
If the testes fail to develop or function during that critical fetal period, the text reiterates the outcome. No male sexual characteristics develop at all. Instead, the default pathway takes over, and female sexual organs form. This really reinforces the principle that the basic genetic and developmental blueprint is female, unless testosterone is actively present during fetal life to suppress female development and induce male characteristics.
What if a boy loses his tests before puberty, but after birth?
This results in a condition called eunuchism. The individual sexual organs remain infantile throughout life, and they don't develop the secondary sexual characteristics associated with puberty. Adult eunuchs, as we touched on earlier, are often slightly taller than average because their bone growth plates, epiphyses, fuse much later, allowing the long bones to grow for a longer period. However, they typically have relatively thin bones and weaker muscles compared to normal adult males. They retain a childlike voice, they don't develop male pattern baldness, and they lack the typical adult masculine patterns of body hair distribution.
And what if castration, the removal of the testes, occurs after puberty, once adult characteristics have already developed?
In this case, some, but not all, of the established masculine characteristics revert or regress. The sexual organs themselves may regress slightly in size, but don't become completely childlike. The voice usually only regresses slightly, if at all. However, there is typically a loss of the masculine hair production. Beard growth stops, body hair may lessen, the maintenance of thick masculine bones is impaired, and the characteristic virile musculature tends to decrease. Sexual desires usually decrease significantly, but are not necessarily lost entirely, especially if sexual activity was well established previously. Erection may still be possible, though perhaps achieved less easily, but ejaculation becomes rare or impossible because the semen-forming organs, prostate, seminal vesicles, degenerate due to the loss of testosterone stimulation, and the testosterone-driven psychic component of sexual desire is greatly diminished.
What can cause hypogonadism other than the physical removal or destruction of the testes? Can it originate higher up in the control axis?
Yes, absolutely. One important cause mentioned is a genetic inability of the hypothalamus to secrete normal amounts of GnRH. Without adequate GnRH stimulation, the pituitary won't release enough LH and FSH, and the testes won't be stimulated to produce testosterone or sperm. The text specifically mentions a condition called adiposogenital syndrome, also known as Froehlich's syndrome or hypothalamic eunuchism. This condition is thought to be associated with an abnormality in the hypothalamus that affects both the feeding center, leading to overeating and subsequent obesity, and the GnRH-producing neurons, leading to eunuchism or hypogonadism. Figure 8111 apparently shows a patient with this syndrome, highlighting the characteristic combination of obesity and childlike sexual organs.
So problems originating in the brain's hormonal signaling centers can definitely cause hypogonadism. What about the opposite situation, hypergonadism, meaning excessive gonadal function, which is usually caused by tumors?
Right. Tumors of the testosterone-producing interstitial Leydig cells are rare, but when they occur, they can produce massive amounts of testosterone. The text says potentially up to 100 times normal levels.
What effect does that have?
In young children before puberty, this excess testosterone causes extremely rapid growth of muscles and bones. However, as we discussed earlier, it also causes premature fusion of the bone epiphyses, so the eventual adult height is actually less than it would have been otherwise. There's also excessive precocious development of all the male sexual organs and secondary sexual characteristics. In adult males, these Leydig cell tumors can be harder to diagnose initially because the masculine features are already present, but signs of excess testosterone might eventually become apparent.
And what about tumors arising from the germinal epithelium, the sperm-producing tissue itself? Are they common?
These types of testicular tumors, originating from the germ cells, are actually much more common than Leydig cell tumors. Because germ cells are pluripotent, meaning they have the potential to differentiate into almost any cell type in the body, these tumors are often teratomas, which can contain a bizarre mixture of different tissues within the tumor mass, things like placental tissue, hair, teeth, bones, skin, etc.
Do these germ cell tumors typically secrete hormones?
Often, they secrete few hormones, according to the text. However, if a significant amount of placental-like tissue develops within the tumor, it can secrete large quantities of HCG, that hormone, which, as we know, has LH-like effects. Sometimes these germ cell tumors also secrete estrogenic hormones, which can lead to side effects like gynecomastia, which is the overgrowth of breast tissue in males.
Okay, that covers hypo and hypergonadism. Finally, let's discuss the very common issue of erectile dysfunction.
Yes, erectile dysfunction, sometimes called impotence, is defined as the inability to develop or maintain an erection that is sufficient for satisfactory sexual intercourse. The text lists several potential underlying causes.
What are the main categories of causes?
They include neurological problems, such as damage to the parasympathetic nerves that control erection. This can happen, for example, as a complication of prostate surgery. Hormonal deficiencies, particularly significantly low testosterone levels, can also contribute. Certain drugs are also known culprits, including nicotine, excessive alcohol consumption, and some types of antidepressants.
But the text highlights that the most common cause, especially as men get older, is often different.
Yes, particularly in men over the age of 40, the text states that the most common underlying cause is often vascular disease. As we detailed when discussing the physiology of erection, achieving and maintaining an erection requires adequate blood flow into the penis and the effective relaxation of smooth muscle mediated by nitric oxide, NO. Various forms of vascular disease, which can result from conditions like uncontrolled hypertension, high blood pressure, diabetes, and atherosclerosis, hardening of the arteries, damage blood vessels throughout the body, including the crucial arteries supplying the penis. This damage reduces their ability to dilate properly when signaled.
So the plumbing itself gets damaged and can't deliver the blood flow needed.
Essentially, yes. The text notes that part of this impaired vasodilation in vascular disease is due to decreased release of nitric oxide, NO, from the damaged endothelial lining of the blood vessels. So the signal for relaxation is weaker and the vessels themselves are less able to respond.
And this understanding of the vascular mechanism is why drugs like Viagra are effective for many men.
Exactly. Erectile dysfunction that has an underlying vascular cause is often successfully treated with a class of drugs called phosphodiesterase 5 PDE5 inhibitors. These are commonly known by their brand names, such as sildenafil, Viagra, vardenafil, Levitra, and tadalafil, Cialis.
How do these PDE5 inhibitors work at a molecular level to help with erections?
They function by specifically inhibiting the action of an enzyme called phosphodiesterase type 5, PDE5. This particular enzyme is found in high concentrations in the erectile tissue of the penis, and its normal job is to break down cyclic GMP, that key signaling molecule that causes smooth muscle relaxation and vasodilation, triggered by NO.
So the drugs stop cGMP from being broken down.
Precisely. By inhibiting the PDE5 enzyme that degrades cGMP, these drugs cause cGMP levels to increase and remain elevated for longer within the erectile tissue, smooth muscle cells, when NO is released during sexual stimulation. Since cGMP is the molecule directly responsible for causing the vasodilation needed for erection, having higher levels of it effectively enhances and prolongs the natural erectile response triggered by sexual arousal.
So it's important to understand they don't cause an erection directly, but they significantly amplify and sustain the body's own natural process when sexual stimulation is present.
That's the crucial point, yes. They facilitate the natural physiological mechanism by preventing the rapid breakdown of the key intracellular signal molecule, cGMP, making the system more responsive to the NO that is being produced.
That's a really clear overview of various potential abnormalities affecting male sexual function, ranging from problems with the prostate gland structure to hormonal imbalances causing hypergonadism originating either in the testes or the brain, and finally, the common issue of erectile dysfunction, often linked to underlying vascular health and treatable by targeting the specific molecular pathway involved in erection. It all links back beautifully to the underlying physiology we discussed earlier.
It really brings home how disruptions at almost any point in this highly complex and interconnected system from the brain down to the blood vessels can significantly impact sexual function and fertility.
There's one final section included in the text that delves into something that's been a bit of a biological mystery. Historically, the function of the pineal gland.
Ah, yes, the pineal gland, deep in the brain. The text notes its rather colorful history, with various functions ascribed to it over the centuries, regulating sex, fighting infection, controlling sleep and mood, even influencing longevity. It's also recognized biologically as a vestigial remnant of a third eye structure found on the top of the head in some lower vertebrates.
Hmm.
For a long time in human physiology, its function was heavily debated, with some considering it non-functional, while others claimed it played important reproductive roles.
But the text suggests its role is now more established, at least in some animal species.
Yes, in some animals, particularly those that exhibit seasonal fertility patterns, the pineal gland now appears to play a clear and important role in regulating their sexual and reproductive function based on the time of year.
Seasonal fertility. You mean animals that only breed during certain seasons?
Exactly. Animals where reproduction is biologically timed to occur only during specific seasons, often ensuring that offspring are born in the springtime or early summer, when environmental conditions like temperature and food availability are most favorable for survival. The text notes that experimentally removing the pineal gland or surgically severing its nerve connections in these seasonally breeding animals causes them to lose their normal restricted periods of seasonal fertility. They might try to breed year-round.
How does the pineal gland actually regulate this seasonal timing in those animals? What's the mechanism?
The exact mechanism isn't entirely elucidated in this text, but it's clearly linked to environmental light exposure. The pineal gland's activity and its secretion of hormones are controlled by the amount and, importantly, the daily pattern of light detected by the animal's eyes. It gives the example of the hamster. In this species, exposure to more than 13 hours of darkness per day activates the pineal gland's inhibitory functions, while less darkness does not. There seems to be a critical duration of darkness required.
And the light signal gets from the eyes to the pineal gland via a specific nervous pathway.
Yes, apparently light signals perceived by the eyes travel through a complex nervous pathway that involves the suprachiasmal nucleus, SCN, in the hypothalamus, the body's master clock, and ultimately reaches the pineal gland via sympathetic nerves, activating its secretion, particularly during the dark phase.
And what does the activated pineal gland secrete in response to darkness?
It primarily secretes the hormone melatonin, along with several other similar indolamine substances. The text suggests that melatonin, or possibly another substance released from the pineal gland, then acts to decrease the secretion of the gonadotropic hormones, LH and FSH, from the anterior pituitary gland. This inhibitory effect might occur via melatonin circulating in the bloodstream, or potentially by it diffusing through the cerebrospinal fluid in the third ventricle of the brain to reach the pituitary.
So, in early winter, for example, as the duration of darkness increases each day, the pineal gland is activated for longer periods, secretes more melatonin, which then suppresses LH and FSH release from the pituitary, thereby inhibiting gonadal function and preventing fertility during the unfavorable winter months.
That's the proposed mechanism in these seasonally breeding animals, yes. This pineal-mediated suppression effectively shuts down reproductive activity during the non-breeding season. However, the text adds an interesting twist. After about 4 months of this suppression, the pituitary gonadotropin secretion somehow manages to break through the inhibitory influence of the pineal gland, even if the long dark conditions persist. This allows the gonads to become functional again, ready for the onset of the springtime breeding season.
So the inhibition isn't permanent, there's an escape mechanism. Does the text reach a conclusion about whether the pineal gland plays a similar significant role in regulating reproduction in humans?
The text states quite directly that the question of whether the pineal gland plays a similar role in controlling sexual drive and reproduction in humans remains largely unanswered based on the evidence presented. However, it does make an intriguing clinical observation. Tumors that occur in the region of the pineal gland, whether they are tumors that secrete excessive amounts of pineal hormones or tumors that destroy the gland by pressure, are often associated with significant disturbances in gonadal function, resulting in either hypogonadal deficient or hypergonadal excessive sexual function developing in the patient.
So, based only on the information provided in the source material.
Based strictly on this source, the conclusion seems to be that while the pineal gland clearly plays a crucial role in regulating seasonal fertility cycles in some animal species, primarily controlled by environmental light cues acting via melatonin, its function in human reproduction remains speculative. The association between pineal region tumors and gonadal dysfunction strongly suggests it might have at least some role in controlling sexual drive and reproduction in humans, but the precise nature and extent of that role are unknown according to this text.
So it remains a bit of an open biological question for humans, fascinating, with tantalizing clues, but not definitively pinned down in this particular text.
Yeah, leaving us with a sense of the ongoing frontiers of physiological understanding in this area.
Wow, we have covered a truly tremendous amount of ground in this deep dive. We started with the intricate architecture of the male reproductive system, didn't we? Mapping that whole network of ducts and glands. Then we traced the incredibly complex 74-day journey of spermatogenesis from a simple germ cell to a highly specialized spermatozoa, all happening within those microscopic seminiferous tubules. We detailed how sperm are then stored and gain motility potential in the epididymis, explored the vital contributions of the seminal vesicles and the prostate gland to forming semen with its very specific chemical and physical properties, including that curious temporary coagulation. We walked step by step through the critical processes of capacitation and the acrosome reaction that sperm must undergo in the female tract to actually enable fertilization, and understood the elegant biological mechanism that prevents polyspermy. We examined the various factors that can affect male fertility, things like sperm count, morphology, motility, the crucial role of temperature, and issues like testicular descent. We broke down the neuronal control and the precise physiological stages of the male sexual act, from erection through lubrication, emission, and ejaculation. We dove deep into the central, pervasive role of testosterone in shaping male development and characteristics, right from the fetus through puberty and into adulthood, and even looked at its intracellular mechanism of action on gene expression. We untangled the sophisticated workings of the hypothalamic pituitary testicular hormonal axis, including its pulsatile nature and the crucial negative feedback loops involving both testosterone and inhibin. And finally, we touched upon the intriguing, if still somewhat uncertain, role of the pineal gland in human reproduction.
Such a foundational biological system, and exploring it at this level of detail, drawing from this text, really underscores the immense complexity and the remarkable precision involved in normal male reproductive health and function. It's quite something.
It really is. Maybe take a moment, as you listen, to reflect on the astonishing coordination required at every single scale here. We're talking from the genetic switches initiating development, and the microscopic cellular machinery building proteins and assembling molecular structures like the acrosome and flagellum, all the way up to the anatomical temperature regulation provided by the scrotum, the pulsatile rhythmic dance of hormones traveling from the brain to the testes and back, the specific enzymatic digestion of barriers by a single cell during fertilization, and the intricate choreography of the nervous system controlling complex physical responses like erection and ejaculation. This whole biological symphony underpins not only the fundamental capacity for reproduction, but also shapes so many of the physical characteristics that define masculinity across an entire lifetime. How does gaining this deeper understanding of the intricate biological underpinnings maybe challenge or perhaps enhance your perspective on male health, development, and the myriad factors that influence them throughout life?
That's a great question to ponder further. Definitely food for thought.
Indeed. Well, thank you for joining us on this very deep dive into the physiology of male reproductive functions.
We certainly hope this exploration of the source material has provided you with some valuable insights and a clearer, more detailed understanding of these complex processes.
We'll see you on the next deep dive.